Welcome everybody to today's Pollination Insights webinar. My name is Rachel McKenzie. I'm the CEO of Berries Australia, and we represent blueberry growers, raspberry and blackberry growers, and strawberry growers. We're very excited about this webinar, which is being presented and hosted by the Australian Honeybee Industry Council, and I'll flip over to Bianca in just a second. But I just wanted to particularly welcome everyone, welcome those of you who are not from the berry industry. We figured that since it didn't actually cost us any more to broaden this out, and it was such a vital issue for the whole of Australian horticulture, if not beyond, we wanted to give as many people as we could the opportunity to join us today. So we're all equal. Everybody is an equal participant. Everybody's questions are valuable. It doesn't have to be related to berries. So just as a few little bits and bobs around housekeeping, there is a Q&A function on the bottom of your screen, and that's what we'll be using for our questions and answers. And you can, the good thing about that is you can pop your question in at any time and we will moderate that here. We do not have the option for verbal questions today, just because we've got quite a few people online. I always like to have a little bit of an intro, so I'm going to use the chat to put my name and where I'm from. And if you feel like introducing yourself, where you're from, which industry, and what you hope to get out of today, that would really help us. But, you know, if you feel shy and want to lurk in the background, that's totally fine too. So I will now hand over to Bianca to formally welcome you as the host of today's session. And I too will be listening with very open ears to hear about the experience from Richard and Ben in particular. So thank you very much, and let the games begin.
Thanks, Rachel. There's two slides from you about who is Berries Australia. I thought you might want to
That's
chime in
right.
and give that update. I mean, I could try my best, but not my
No, that's fine. I
expertise.
think it's not particularly exciting, and I think I've covered off on most of it, which is that we're obviously a joint venture. And we do like to say that we're the largest fresh produce category, although our vegetable friends might argue around that definition of what is indeed a fresh produce category. And we do have pretty good representation, particularly across the blueberry industry. So what's the next slide, please, Bianca? Oh, I just thought this was a little bit interesting in terms of the Varroa response. I'm sure you're all aware that we tried very hard to eradicate it and we were not successful. But as industry associations, we lobbied very hard to have the response categorized to a Varroa 2, categorized to category 2, which saved the berry industry around 30%. And I think it's just really important when people understand how the contributions were done, and you can have a look for your own contribution. But for example, the berry industry, we contributed approximately 1.6 million to the response. So, you know, we've all paid our share, and unfortunately we weren't successful, but, you know, it's really important that we continue to work together to deal with Varroa. And whilst he's not here today, I think it's really important to acknowledge Danny LeFeuvre, who's the CEO of AHBIC. He did an amazing job in this space, and I think he gave up his life for about 2 years for Varroa. So I'd like to, you know, make sure that we publicly acknowledge him. Okay, now over to you, Bianca.
Lovely. Thank you so much, Rachel. And thanks to all of the participants. It's wonderful to see so many people online. And I think this is going to be a really valuable webinar in the future as well. So I work for the Australian Honeybee Industry Council. We're the national peak body for the honeybee industry in Australia. We're a federated model, and we have representation through our member bodies, which are associations across Australia, which make up a number of different places in the country, and also some national associations in terms of queen breeders, pollination, crop pollinators Australia, and amateur beekeepers as well. If you just go to the next slide. So what are we here for, and how come we're all joining in today? And I think the number one factor is all about Varroa. So I've highlighted the states in which Varroa has either well and truly spread itself far and wide, or has begun to really take the capacity of the beekeeping areas of those states. We've generally had Varroa here where I'm from in the Lower Hunter since about June 2022. So I'm in New South Wales. There's been some really unique experiences coming out of the New South Wales beekeeping industry, both commercial and recreational. It's really important to remember that European honeybees are not the main and initial host for Varroa mite, so they've transitioned across into the European honeybee populations. Australia's been one of the last large agricultural and honey producing countries in the world to actually have Varroa established, which is an interesting concept for all of the pollination dependent industries. In terms of what happens to the colony, they'll suffer an immense compounding amount of stress from Varroa and other honeybee pests and diseases. And when this goes unmanaged, the honeybee colony will eventually collapse and die. And this goes for beekeepers who don't manage Varroa and also for feral colonies in the environment that are not managed at all. And this has been the case across the globe where Varroa is established. So beekeepers now don't just need practical and physical abilities, business management skills. They have to have an intimate knowledge and a continued understanding of the needs of their colonies to keep them going. And what we're seeing across the areas in New South Wales where Varroa has been established for some time is really an increased loss of feral bees. And that's adamant. It's coming through from lots of different people. Next slide for me, please. The feral honeybee population will be further discussed by our Professor Ben Oldroyd online, but I just wanted to note that the cost to manage colonies by beekeepers is rising because feral colonies are disappearing. We're looking at the opportunity for beekeepers to become more dependable for pollination-dependent industries. There'll be increases to the pollination demand as a result. So no feral bees in the environment will mean no feral bees available to do that free pollination that we've all come accustomed to in Australia. There will be, and there has been already, reports of bee businesses that have come up for sale as a number of beekeepers are expected to exit the industry. Somewhere between a 20 to 30% reduction in commercially registered beekeepers across Australia over the next 5 years, which is quite significant considering the scale of agricultural production that depends on honeybees. Next slide. As we know, the pollination by honeybees is a critical aspect of not just berry production, but a lot of pollination-dependent industry production in Australia and around the world. Our honeybee industry and subsequently the pollination-dependent industries will rely on the public lands the way that beekeepers do. So we use public lands and native flora to build strong hives in the seasons when we're not pollinating crops through pollination contracts. It's this balance of the two that needs for crop pollination and the foraging of native flora for making honey as a paramount consideration into the future for both our beekeeping industry and your plant industries. So the native flora for building bees is a really increasing scarcity for beekeepers. And because of this dependency and the way that a colony functions, it's important for berry growers and other growers of other dependent crops to just understand the importance of providing an environment in and around crops that nurture the honeybee colonies that they have on site. And it's invaluable to recognise the critical aspects of beekeeping to help foster mutual benefits between growers and beekeepers into the future. A little bit about what Varroa destructor will cost bee businesses. So predominantly time management and cost are the two main factors. In some recent surveys that we've provided through industry, time management has increased and costs have increased around 30% to the beekeeper. And that could be a result of the extra time that it takes to manage Varroa with chemical IPM or integrated pest management steps. It also costs money to purchase chemicals, but it also, in addition to managing Varroa itself, it's also the loss of colonies, which is a factor in the future of the beekeeping industry. Beekeepers may actually choose to employ more staff to compensate for the extra workload that's involved, and they'll also reduce a little bit of the time of their colonies being productive if they've lost colonies by reproducing nucleus colonies in their systems of their businesses. And this can also cause a reduction in honey production in some cases because of the way that colonies need to build up over time and be ready for effective pollination service. So that's just a small snippet. There is a lot more to it, but I would like to really focus now your attention onto Emeritus Professor Ben Oldroyd from Sydney University. Ben's going to share some really fabulous insights about feral bees and all of the things he knows. Thanks, Ben. Take it away.
Thanks, Bianca. Um, okay, thanks for the first slide. Um, I've been interested in feral bees for a very long time. Started in the 1990s, and we'll come to that in a moment, my first experiments. And I'll be giving a very brief summary of what we've been doing. And then I'll end up talking about what Bianca just talked about. How will the feral bee densities change now that we have Varroa? And then a few ideas about what the consequences will be for pollinator dependent industries such as the berry industry. Okay, next one, please. So as I said, this story started in the 1990s. I left Australia in the 1990s and went to the USA and worked there for 5 years at the big bee lab in Baton Rouge, Louisiana. And then I came back and started this project on feral bees in Whitefeld National Park. So apologies for the rather old-fashionedness of this slide, but it still tells the story that you need to know. There's some shots of Whitefeld. It's basically a heathland, which is an old seabed, and it's got a creek that runs up it, which drains the Wimmera River. We used to drain the Wimmera River before we used all the water. And it flows into this thing called Outlet Creek that used to flow into the Murray at Mildura, but no longer does so. Water doesn't enter the park anymore. So along the creek, there are various now dry lakes, and they're surrounded by beautiful red gums and things like that, black box, which you can see in the top right. And it's a fabulous place for bees. It's just great honey area. Beekeepers used to go there. There's an old thingy there on the bottom right, which is where old-fashioned beekeepers used to hang their colonies up to keep them away from ants. So I was looking around for a site to study feral bees, and National Parks Victoria told me, go to Wyperfeld. And the reason for that was that feral bees were so dense there that they were harassing tourists and potentially competing for tree hollows. Well, I won't be talking about tree hollows today, just about the feral bee population. Can I have the next slide? Yeah. So we went there every 3 months, or 4 months, sorry, every 4 months for 3 years. And National Parks managed to organize this group called the Victorian Trust for Conservation Volunteers. And as far as I can tell, these were all wayward Swedish youth that had been sent to Australia to volunteer in a conservation project. So that was interesting, wrangling these kiddies from Sweden. So what we did was we set out these plots, which you can see in the, well, can you go back to the previous slide, one before this? Thank you. You can see the plots 1, 2, 3, 4, 5, 7, 6, if you go up Outlet Creek. And there we had 500 meters of 100-meter square plot. So 100 meters, 100 meters, 100 meters, all lined up in a row along that red gum fringe of the creek. So next slide, please. This is the result. So we've got the number of colonies per square kilometre, and you can see it's extremely variable. It peaked at about 153 colonies per square kilometre. So we are walking around the trees in this plot. We're looking at each of them from 4 angles, north, south, east, and west, counting the bee colonies that we could see in the tree. And I think, at least after a while, it got very accurate because we knew each individual tree by number, if not by name. And we knew all the hollows in them, and I think we found every colony. So 153 was the maximum and got down to about 50 in the middle of the drought. We'll go to the next slide. So that's the key take-home message. Feral bee densities can be extremely high, but I just want to put a caveat on that, which is that that's just along the perfect habitat in the red gum forest. Out in the dunes of the old seabed with banksia and things like that, it would have been much less because colonies can't live in those little trees. So this is a representation of the 5 plots, 500 metre by 100 metre square plots at the 7 sites. So that's basically the 7 lakes. And squares are, which is which? Circles are established colonies, and squares are new colonies, new swarms. So you can see it was very variable, but you might have noticed, the canny ones amongst you, that they're aggregated. You can see that they're in group, in clusters. So it's not an even spatial distribution, it's a clustered one. That's interesting for spread of diseases, that they seem to hang out together and their swarms are attracted to where existing colonies are. So that's a interesting observation. That probably means that Varroa will spread quite nicely in the feral population. Okay, so that's okay, walking around and putting labels on trees and looking at them. And if you've got enough wayward Swedish youth, that's probably sufficient. But it's difficult and really expensive. So if you look at that and you say, I've got to survey every tree and count the feral bee colonies in it, it becomes too daunting and too expensive. So we turned our mind to alternative ways of assessing honeybee colony densities at landscape scales. And there's a potential solution, which is to use the reproductive biology of honeybees to estimate the density of colonies in the environment. And in order to explain this to you, we need to talk about bee bonking. So let's have a look at a video of bees bonking. We hope. Yes. No. Oh, well, looks like it's not going to work. Oh, here we go. So these are the drones chasing a queen, which is going to a drone congregation area. And they mate on the wing. Queens mate about 30, 40, 50 times on one afternoon, once in their life, and then they go back and lay eggs for the rest of their life. After the drone mates, he turns inside out like a rubber glove and falls to the ground and dies. Plunk. Okay, so now you know how bees mate. Have the next slide, please. So they do this at specific places. This is one which I discovered when I was an undergraduate at Sydney Uni in 1978. It's #1 oval at Sydney University. And if you go out into the middle of this oval and toss a stone into there, you'll see drones trying to mate with the stone. So they're quite common in the landscape. They're usually an open grassed area surrounded by trees. Suburban and rural football fields are ideal locations for a drone congregation area. So you go to the drone congregation area at the mating time, which is between 2 and 4 o'clock in the afternoon, and you put up a helium balloon with cigarette butts painted black, don't forget the black, hanging underneath the balloon and a trap. Drones will try and mate with them because you've been clever and you've inoculated them or treated them with queen pheromone. It's a sex attractant. The next slide, please. So this is a video of using the trap that we developed. Those are the cigarette butts. And we're going to put queen pheromone, which is 9-oxydecanoic acid, onto the cigarette butt. And this becomes irresistible to the males, as you will soon see. There's the males going into the trap. They go up to the cigarette butt and they go, oh, this isn't a queen, and they break off and fly up into the trap. And at that point, you can catch them into alcohol. And then you've got a nice bunch of males, presumably from all of the colonies in the area, that you can analyze genetically to estimate the number of colonies that are out there. Next slide, please. So the principle is, if we've got a feral queen heading a colony and she's diploid, so those numbers there are just a label for the genes, the alleles at a particular gene, or locus, as we like to say. And she will lay unfertilized eggs, which produce the male honeybees. Don't worry about that. That's just what bees do. And so you'll get Half of her males will be that column you see on the left there, 22, 36, 13, etc. And the other half of the males that she produces will be 28, 36, etc., etc. So all you've got to do is count the unique haplotypes in your balloon trap and divide it by 2. You've got an estimate of the number of colonies in the trap. Okay, now it's a little more complicated than I've stated, but you get the idea. Okay, next slide. So what we can do is we catch our drones, we genotype our drones in a big expensive machine that goes ping, and then we divide the number of haplotypes by 2, and that will tell us the number of feral queens in flying range. Next one, please. So in order to convert this to a density of colonies per square kilometre, we need to know the distance that the males fly to mate. By the way, that's a drone trying to copulate with a cigarette butt. We spent about 5 years doing this research, figuring out how far males fly. Some estimates, previous estimates, were something like 16 kilometres to a couple of 100 metres. So what's the real number? The area that you're sampling. And the next slide, please. So this is the skeps there represent colonies in the forest near where you're sampling. And in this case, if we said that you found colonies, 3 colonies in your trap, that would be 3 colonies per 3.14 square kilometers, right? The 3.14 is pi. So you're going pi r squared. The radius is 1. So it's 1 squared times 3.14. And then there's 3 colonies divided by the number of square kilometers. You get 1 colony per square kilometer. Okay. If, on the other hand, the males fly 2 kilometers, you'd be sampling, 6 colonies there, and you'd get a different estimate. So I actually had worked that out for you. Let me see if I can find it. Yeah, so in this case, it would be 0.5 colonies per square kilometre, so half what we saw in the previous example. So we needed to figure out how far drones fly in order to mate and how far we would possibly catch them from their nest. So this is a very cool experiment, which I'll tell you in a sec. Have the next slide, please. Yes, 0.5 colonies per square kilometre. So what we did was we jazzed up a colony at Sydney University. So we started in July, feeding it pollen and sugar and water and really making it produce large numbers of drones. And then we took them over the Great Dividing Range to Lyndhurst, which still had frosts at that time of year. So the local bees weren't producing drones, but our colony was a big booming one. And we paint marked 3,000 drones inside that colony just with a spot of paint on the thorax. You can see them coming out of the colony on the right, bottom right there, a red one and a white one. And then, as you can also see, the kind of countryside that it is there. It's mixed cropping and grazing in Lyndhurst. Okay, next slide. Okay, so that's an aerial view of the place. The hive is in the middle. And next slide, please. So there's the hive. We went 200 meters steps away from the hive and figured out when was the last location that we found a marked drone. Okay, that's the distance that the drones fly from the hive. So this was the last drone we found, and that was, ta-da, next slide please, 3.75 kilometers from the hive. So it's quite a long way, and you sample quite a large area. So that means that we're sampling an area of 44 square kilometers. So we need to, once we've done our genotyping and found the number of families in the trap, divide that by 44, and then you've got the density of colonies in the area. So using that, what have we found out? Can I have the next slide, please? So we've only done this properly 3 times. We did it before when we thought the area sampled was much smaller. So, but this bit you can believe. So we did one study at Wagga in mixed cropping and grazing. We found 236 colonies in the trap, which equates to 5.4 colonies per square kilometre. Lyndhurst, we've got a lot of other drones in the trap, so we were able to genotype them too. That was 6 colonies per square kilometre. And then the record so far is in outer Melbourne, which was kind of suburban and small crops, which was 7.3 colonies per square kilometre. We also found out that you need to sample about 3,000 drones before you get a decent estimate of per colony density. You get some weird statistical things happening if you have smaller than that. We found much higher densities of colonies in undisturbed habitats than in cleared habitats, which I think Bianca alerted you to that earlier. Here's the thing. Feral colony numbers in Australia in the bush are high, but they are about 1/10 of the recommended stocking rate for most berry crops. So 6 colonies per square kilometre is 0.6 per hectare. So that's not going to be good enough. Ferals are probably sufficient, around that 1 colony per hectare for apple, pear, canola, things like that. So if they're right in the bush or right next to natural bush, now prior to Varroa, they would probably, they'd take some yield hit, but it wouldn't be, severe. And the final thing is that ballooning is also great for bee surveys and finding out whether you've got exotic diseases in feral bees. Just catch the drones and genotype them. And that was used during the Varroa incursion. So what will happen now? Well, I think Richard probably got more to say about this than I do, but the experience around the world is that there will be a reduction of feral colonies of at least 90% and probably all of them in the short term, and then they'll start to build up as resistance is selected for by natural selection. That means that most crops requiring pollination will now need to rent colonies in higher numbers, and they'll have to pay more for them because there'll be more effort to prepare them for the pollination contract. Also, honey production may rise because the number of colonies in the bush is higher than is recommended for honey production. Okay, so they are the ferals in the bush at the moment are at higher densities. It's a density that would suppress honey production if you were in a commercial situation. Next one, please. That's it from me. I thought it was me, the end. So thank you for listening, and we'll take the questions at the end, I assume.
Yes, wonderful. Thanks, Ben. Yeah, we will definitely be taking the Q&A to the end of the webinar. And just a reminder to the listeners online, please, if you have queries, pop them in now. We can collate them towards the end, and we'll run them through to both of the presenters at the very end of the webinar. I'd like to hand over now to commercial beekeeper and kiwi fruit grower, Richard Klaus from New Zealand. So welcome, Richard. Thank you so much for joining us.
No, you're welcome, and that's I hope you can gain some information out of it. Now, if we could go to the next slide, please. Yep. Is that all good? I've got my secretary helping me today, my daughter. Life after Varroa, their experience in New Zealand. It was a challenging transformation, but I think we're slightly better beekeepers for it. And as far as orchardists goes, we're a lot more aware of what's happening in our orchards. And just to put a little bit of relevance in what's happening with Varroa, Varroa is about the size of my hand on my body, that's sucking blood out of my system, continuous while you have Varroa. So you can imagine when that's happening to a bee, if it has if it caught a cold, normally before Varroa, it would now be the flu. So everything is sort of tenfold more. All the diseases are more prevalent. It's weaker. The immune system drops. Yes, not a nice life for a bee. I've been a beekeeper for about 30 years. I've been involved as a zespri auditor since 2009. So it's had a lot to do with the pollination side. Now, the brief background of Varroa New Zealand, it came here in 2000, April 2000, and we had the same problems with what you're having in Australia with the feral bee population actually really reinfecting continuously the managed hives. As far as a grower was concerned, well, for us on our orchards and fellow growers, we got a lot of free pollination from the feral bees, and then all of a sudden they were vanishing. And so we had to be quite aware that basically the free ride was over. We've got to look at our orchards, whatever we're growing, realise what we need in the number of bees at a certain strength. What do we need as the maximum amount of bees to grow our crops? So when we do look at crops, if we look with our kiwi fruit, we have about 7 to 10 days to pollinate the kiwi fruit. So we would look on the worst case scenario, as in if it's going to be a wet period, we would look at the higher number of hives that we need so it's successfully pollinated. The impacts on feral bees, unfortunately, we don't have feral bees any longer. I've heard a lot of people have discussed this here about you get certain traits in bees that they can start to live with, Varroa. We call it the hygienic trait. Varroa just keeps tipping them over. Hopefully it will happen one day, but we need to have a lot more research on that. Then the impacts
Hey,
to
Richard,
pollination.
sorry. Do we need
Yes.
to move through some slides here?
Oh, yeah, sorry. Next slide, please. Beg your pardon. So Varroa's initially put a lot of pressure on beekeepers and beehives for the first few years while all this, death rate was happening with the feral bees. And then when it started noticing in orchards, some of them started having inconsistent levels of pollination, misshapen fruit, etc. And then if it's a little bit of work done by MAF or MPI about what we actually need for the correct number of bees per hectare per crop. With the kiwi fruit, we then started introducing superior pollen producing males to increase the viable pollen available in the orchards, and that's enabled the bees to actually transfer pollen far more efficiently and get the better crops out of it. Next slide, please. Then moving forward, unless you rent or hire beehives for pollination or have your own bees, there's a good chance you will not have bees around your orchard. As a grower, you'll be competing for pollinating hives against honey production hives. You need to start having a discussion with your beekeeper. We now set up hives quite differently to what we used to beforehand, because we originally just targeted honey hives and would pull them out of honey, put them in the kiwi fruit or avocados, and then put them back into the honey, and life carried on. But now for different parts of the season, we'll have hives set up at different strengths. So that's something you really need to start addressing with your beekeepers to see what's available at that time of the year and what you need to pollinate. Like, i.e. early in the season, you'll need a smaller, like a nucleus hive or a growing single box hive. Later in the season, you'll want a 2-box brood hive that's growing into quite a strong hive, ready for honey pollination.
Excuse me,
next,
Richard.
yes.
I'm conscious. Sorry, Richard. We've got a lot of avocado growers on the call. Or avocado representatives, do you know what the you just use blueberries and kiwi fruit? What do avocados have to do? Oh, if you know off the top of your head, if you don't worry.
Yeah, no. We haven't pollinated avocados for quite a few years because of the spraying issue. Why the bees are in the avocados, but it's the same thing. The avocados, I think from memory, need around 6 to 8 hives per hectare, and a lot of those hives, we call them a growing hive, so they'll be around 12 frames of bees in total, and around that 3.5 to 4 frames of brood inside them, so they can grow while they're in the avocados without swarming. And yeah, so you will, well, we find in New Zealand anyway, if you don't have bees in them, you'll struggle for pollination. Like flies will do a lot, but bees are still the primary source of pollination. If that helps sort of cover that a little bit.
Thank you.
Yeah. And so in the North Island, as far as Varroa spread each year, and it was transferred an awful lot by man moving bees, for honey production out of the Manuka honey in the Manuka heyday. So it transferred around the North Island very quickly. And within 2 to 3 years, your honey production went up. But definitely you weren't seeing the feral bees any longer out, pollinating around these bush areas. So it's what we now have classed as a feral bee is swarms that are coming from man-managed hives. And we are finding that they don't live very long, probably past June, July of the same year. A few of the canolean bees will live until August, but by then they're pretty much all dead nowadays. Do you use bumblebees a little bit in Aussie?
They are only endemic to Tasmania, and they're actually considered a feral pest in mainland Australia, which is a very contentious issue.
Yeah, okay. I'm surprised with that. They do compete with honeybees, where we have the whole 5 species on an island. It's a quarantine island, but we have 2 on the mainland. We have been experimenting the last few years of using them now in glass houses, tunnel houses that are quite enclosed because they seem to orientate better than the honeybees.
Mm.
So it's certainly a factor to investigate. And they do survive far better in that, and I'll explain that a little bit later in the presentation. Next slide, please. Yep. So the case study I was using was the green kiwi fruit because it's the only kiwi fruit variety that was commercially grown before Varroa arrived and after. And before Varroa arrived, we could have 6 to 8 hives a hectare with quite variable male plants of lower, medium to lower quality pollen viability, and would still get pretty good, consistent pollination results. After Varroa, we now increase our hive numbers to 10 to 15 to the hectare, and we've now introduced some very good quality producing males. And that's now letting us in this factor without the feral bees now of getting every flower visited by bees will be well pollinated and a higher chance of being pollinated right through the orchard. So it's a little bit in there with your different species of plants like avocados. I believe you do the same there. You have pollinators inside your orchards. Just to get to know your orchards and give your orchard the best chance of being pollinated. Then the early signs of Varroa is inconsistent yields. In our kiwi fruit, we notice misshapen fruit. There's been a little bit's been said before about the taste in fruit, the dry matter ratio. We've certainly noticed that also. And just be a little bit cautious because it has happened to us. You might think you've got a lot of bees in your area that are still doing pollination. You think these are feral bees, but just over the hill where you can't see, they may be man-managed hives. And if they are removed to go to an area for honey or go somewhere else for pollination overnight, while your orchard's flowering, you may not have any pollinators left. So hence, you need to be in touch with your beekeeper and have hives available to pollinate your orchard. The stocking rates, the experience has been you do need to increase your stocking rates at certain levels because obviously the feral bee population is gone for us. And also with your stocking rates, have a look outside your orchard or the farm you're growing the stuff on and just look what's competing for floral sources. You might have, you've alluded to before about flowering gums, could be clover paddocks, etc. And if that's the case, you may need to allocate a higher number of bees for your pollination. And this is just what we run off a little bit here. With MPI on the left, so the blackberry boysenberry raspberries, you'll probably have similar hives per hectare over in Aussie. And so if we're looking at a sheet like that, like let's say the blueberries, 1 hive per hectare, if we've got a lot of clover, it seems to be when our blueberries are flowering, some of these guys are quite often putting 4 and 5 hives to the hectare, just so you still, with that competition, you still keep enough bees in your orchards. On the right-hand side, this was reviewed in 2018. Kiwi fruit aside, because of PSA, we've had to put higher levels of bees in. You'll see the different stocking rates that we have for different crops. But some of these crops, like our berry crops, are earlier, so we'll have a smaller hive. It's going to be a growing hive. PSA is a bacteria that gets in and kills kiwi fruit. And it's quite a nasty little thing. Now, changes the farms in our orchards. There's a few things we've had to do. Is we want to make things so bees will stay in our orchard nowadays. So we have a nice available water source for bees. A dripping tap on concrete seems to be the best because the bees can get on the concrete, drink the water. Plastic buckets are not ideal unless you sand them with sandpaper because the bees will slip into the water and drown. Orchard structures need to change slightly, and this has been quite a struggle in New Zealand because we've got quite a disjointed beekeeping industry. I think you're a lot luckier in Australia. We have like tunnel houses, clear plastic. We have a lot of white wind cloth in orchards and a lot of white hail netting also in the orchards. You'll see a graph there with bees and humans. The bottom one is what bees can see in their vision, and the top ones is to put it into what we can see. So they can't see black, they can't see white.
Oh, great.
So, yeah, so if that's of any help, and so with this, we've got one company in New Zealand started doing it. I helped do the trials on it about 20 years ago, and this is how slow the uptake is. Actually, on the bird netting for overhead, and it's also for the bird netting for the sides, it's a blue and white weave. When you go back to the colour chart, bees can see blue. And so when we put that over our side of bees, within 20 minutes, they were working equally as well or better than the control apiary site. So just something to think about moving forward in the future. If you have a completely white overhead roof and white sides, your bees will disorientate because they can't see the sun, they can't see white. You'll lose a lot of bees, but for an orchardist, it's actually our foraging bees. So we're losing all our foragers that do our pollination. Then when the bees leave the orchard, we have an inferior pollination, but then as a beekeeper, my hive's now compromised for a honey crop. And then am I going to come back and pollinate your orchard or not next year? Yeah, that's the question. And that's just why I say you're very lucky you've got that good beekeeping industry set up in Australia, and you can use it for discussions like this here. There's a website you can go onto and you can just look it up and they'll show you what it looks like, and then you guys can discuss it from there. So is life under Varroa manageable? Yes, it is. As a grower, our understanding of pollination requirements in our orchards, we're now walking in there with our eyes open, and we're more aware of outcomes that we've been making in the orchards. And we've become more informed too about what's happening with the pollination needs, like in our particular orchard, and we encourage a lot of people to do it. There's more and more doing it now as with kiwi fruit pollen's very low nutrient level. I think it's missing some essential amino acids, which is getting tested this year, finally after 20 years of asking. And so we recommend don't mow your orchard floor, leave all the yellow flowers on there because they have a higher fat content and other nutrients. and the bees pollinate longer and better. So it's just small things like that. Be aware, walk in the orchard and have a good look. As a beekeeper, um, pollination has now become our primary source of income because it's a guaranteed source of income whether we have a honey flow or not. Honey is now rapidly, because of the cost of production, becoming supplementary now and obviously varies year on year. And we are having beekeeping businesses that only do pollination now. They put their hives out to make 1 box of honey for winter food, and they just leave them like that. With the honey on, the bees keep healthier than sugar feeding. So, that's a positive. So it can be a positive change for both growers and beekeepers. For us, it was quite a radical change because no longer you could just do honey and survive on it. But communication between growers and beekeepers is absolutely essential. You've got to communicate with one another, your good things and your problems. And the next slide, please. So another thing is, the biggest thing to realise, it may take some time because you've got such a big country, but free pollination is going to be history. Once Varroa goes through your feral hives, I think that's pretty much it. You want to make your crop, and I think this is really, really important. This is something the commercial growers in New Zealand struggle to get their heads around. You want to make your crop a crop of destination of choice for myself as a beekeeper to come to first. So just the little things in there, like, you got the right plants in there for pollinating your orchard, water, your overhead shelters, etc. So when the bees turn up and they leave, they're in equal or better condition. Be prepared to pay for your pollination services. A pollination payment is just a compensation payment to a beekeeper for the loss of honey production and his time moving the hives in and moving them out of your crop. Have a pollination contract. This is really, I believe, is really, really essential. And this sets out the requirements for both parties. And I could send you one over if you require, that we have for the, specifically for the kiwi fruit. And then this needs to cover the whole list of things, like the number of hives per hectare, time of delivery, the required strength of hives, what happens with spraying, orchardist responsibilities, the beekeeper responsibilities, duration of pollination. There's a whole list of things, and it's a really good document that you need to have, and then everybody knows what their responsibilities are, because now you don't have a never-ending resource. And then just start planning now while you still have time, because it sounds like you still do have a reasonable feral bee population. So start planning now for the absence of those pollinators. Talk with your local beekeeper and start creating a pollination contract and get to know your orchard. That's really important. Really get to know your orchard, and things like your bee-friendly cloth, orchard structures. You might, I've been involved with Cesbury this year. We've done a pollination roadshow, and this coming season, we're hoping to do some more research on these covered orchards and markers, lifting sides, etc. And it's really important for both parties that we get this thing right. But yeah, just communicate with your beekeepers, have a good look in your orchard, and just start doing these things. I hope it's been of some use, and thank you.
Thank you so much, Richard. I'm going to ask a question to get the
Yes.
ball rolling. So at the moment, we encourage our growers to grow plants near orchards that attract bees, but it sounds like in the future that will be actually competition. Is that true?
No, it depends how many plants you're planting. If you're planting enough so they can supplement their diet in your orchard, and it's actually be a good thing. And I'll just give you a quick little idea, like kiwi fruit pollen. The hives this year on average made 2 frames of white kiwi fruit pollen per hive. We went over 5 days later to lock them from a 2-box brood after we moved it from the orchard into a single box brood to set them up for honey production. And there was barely one cell with white pollen left in it. It was all pulled out the front door and chucked on the ground because it's missing something in their diet. So if we could have something in their diet that supplemented that, that wouldn't be all that wasted energy and wasted resource. So no, I think within reason, absolutely. Like if you've got buttercup on your orchard floor, a little bit of the purple flower we get, yeah, just I think anything for variation of diet, because bees don't harvest one form of pollen at one time. They harvest multiple sources at one time because they've done this for millions of years. They know that one of those pollen sources will be deficient in one of the essential amino acids, so hence they harvest the others to mix together and compensate. Yeah, so no, definitely I would agree is that plant something, but you don't go and if you've got a 1 hectare blueberry orchard, you don't plant 10 hectares of flowering gum, for example. You might plant 10 flowering gums. So it's just supplements.
Right, thank you. Bianca, there's a couple of questions, but are they from you
Yeah, I've put some common queries that I'll ask at the end, just to, if we have a bit of time, just to add to the volume of information that Richard and Ben have both shared.
Right.
Sarah has asked or made a comment, I guess, in the chat that says she'd like a copy of a pollination contract. And there is some more information coming up from me as I delve into some points and key areas of the pollination guide for berry production. So
Yeah. Well, maybe we can jump into that because it's only a 1 hour webinar, isn't it?
That's right. Yep.
Got 7 minutes left. So let's get
Yep.
into that. Oh, look, again, I think this is on our website. So whilst it is a pollination guide for the Australian berry industry, it does have application beyond the berry industry, and in fact was adopted from the almond berry pollination guide, and it is on our website. If anyone who's not a berry grower doesn't have it, or if any berry growers don't have it and want a hard copy or need to access it, please let us know.
Fabulous. Thank you so much. Look, Richard, that was fabulous. I think you've really covered over a whole bunch of things that are in practice now in New Zealand that we can only hope Australia takes note of. And I think with this pollination guide for the berry industry, as Rachel said, it's not just a one-stop shop for berry growers only. It is absolutely a resource for many, and it can be interpreted in many ways across different horticultural industries, along with other information that's already been conducted by Hort Innovation Australia and other places like AgriFutures Australia in the honeybee pollination areas. So I wanted to just touch, and I'll be really quick. I'll run through these as quickly as I can without skipping too much, noting that the webinar will be recorded and that the guide is available online. If you skip to the next slide for me, please. The big point, and I think Richard really touched on this a lot, is communication. It is absolutely key. It's the first step in a successful future for berry growers, horticulture in general, with the beekeeping industry. So yes, there is competition between when beekeepers need to have hives making honey, which also produces healthy bees. About 75% of Australia's honey is produced in native forests, and about 45% of the time beekeepers are working hives is when they're making honey. So we're balancing those times in the industry together into the future to make sure that all of our practices can align to make a better future for everybody involved. Next slide. There's lots more information in the guide, but just a couple of points about pollination and grower communication. So we really want beekeepers and growers to chat about the pollination timeframe for each of the growers. I know that many berry growers might grow different types of berries, and they may all have different types of flowering opportunities. Those conversations are key at the beginning of these spaces where we're starting to negotiate a little bit further with industry of beekeepers to make sure that the timing of all of those things line up in agreement with all of the other components. Definitely pollination agreements. There's definitely room for those to be implemented if they're not already. Double checking that growers, beekeepers that you utilise are compliant with the Code of Practice, the National Beekeeping Code of Practice. Looking into independent third-party hive health auditors for some access to information about what those beekeepers are providing you in the colonies, because not all colonies are the same, and having effective pollination depends on appropriate hive health. And also having a good discussion and sharing a pesticide plan and IPM plans that you might have on farm that could coincide with IPM plans that beekeepers have now and moving forward, especially with Varroa. Hive standards is the next slide, please. Very important. Richard touched on this. In Australia, we do have some specific details in this guide around what beekeepers should expect to be able to provide if they haven't already engaged in working with growers in the berry industry or other industries. A really good conversation around what hive strength and standard is appropriate for the crop that you are pollinating. It's really important, and I really urge people to jump in and review some of that information and talk to beekeepers and reach out to even to AHBIC to get more info. I'll click to the next slide just so we can make sure we cover all of the pages I've put together. As we mentioned, there is a copy of a pollination agreement template. It's quite a few pages long at the very end of the pollination guide for berries. It's a fabulous component. It really does step you through the number of hives, the price, audit arrangements that if you do engage an independent auditor, the timing of colonies coming in and out of the orchard. And this is really important as well if you're managing different pesticides, if you have an integrated pest management process, or if you've got staff on site, if you've got other components of on-farm activities. Pesticide plans are really part of this process. We don't want our bees to come off a pollination contract in a more negative state than when they went on. We'd like to try and make that balance quite equal between the beekeeper and the grower themselves. Clean water source. This is a common point that beekeepers make about access to clean water on site at all of those pollination events, especially in netted crops as well. These are really important topics that are really spelt out through this agreement, and it's a fantastic place to start. Even if you are not a berry grower, the pollination agreement template in this book is really quite extraordinary, and it's a great starting point for other industries to have a look at in the future. Now, next slide. We have a National Honeybee Biosecurity Code of Practice. It was recently reviewed and updated to include Varroa. This is a really important document for beekeepers in Australia, and we encourage all beekeepers to abide by the details throughout the code, which includes looking at beekeepers managing colonies, managing pest and disease, the number of times they might implement a change to their practice to manage a pest or disease. All of the necessary components of really good, healthy beekeeping are included in this code of practice. There's lots of beekeepers that can turn up on sites when pollination contracts are underway, and a lot of different crops in similar areas will demand that beekeepers sort of come to the site, but that also increases the risk of biosecurity concerns with the density of crops being pollinated at one time, the movement of hives in and out of areas across the country to meet the needs of pollination does increase the risk of biosecurity concern for both the farms that they operate on and the honeybee colonies that visit from time to time. So unmanaged biosecurity risks are pretty important to be aware of, and we discourage that so that good beekeepers can provide really quality pollination services into the future time and time again. It's developed to incorporate fundamental biosecurity principles, and we really want beekeepers and growers to understand their requirements following that code. Next slide, please. We did talk a little bit, Richard touched on some information about what it means to actually care for bees on farm. There's some really fantastic documents that coincide with the guide. At the end of the guide, there's some links to some bee-friendly farming, but generally I suppose growers are not beekeepers necessarily, and we want them to understand that there are really significant impacts on hives that will determine the effectiveness of pollination. Cold weather, nectar and pollen availability. So not all tree and plant species produce the same quality nectar or pollen, or both in general. So insufficiencies in either of those can have follow-on effects that relate to hive health. Extreme heat can be an issue for pollinating honeybees. It can also be an issue for non-honeybee species. So some of our native bees can be very affected by extreme heat conditions. The location of hives is really important. There's further information about where to put hives on farm if you're not doing that already. And I know that a lot of berry growers already have some of this knowledge, but if you wanted to brush up on it, highly recommend checking out the Caring for Bees on Farm section. Covered production has got some specific information that is different in the hive management to address the timing of hatching of brood especially. So there's some really key components in the guide about that. And again, communication is key to being informed as a grower of what the beekeeper can achieve and what their expectations are when they're on crop. Next slide. Insecticides and fungicides and surfactants really do affect bees. Understanding what chemicals to avoid and how to use chemicals wisely on farm will ensure that the safety of honeybees before, during, and after a pollination event are managed well and promote the opportunity for beekeepers and growers to have a fantastic future together time and time again. Systemic and contact insecticides can be potentially carried back to the hive in the nectar, in pollen, and in water that's collected by bees. So if insecticide residues are attached to pollen and brought back to the hive, they will be fed to the bee brood, and it may result in deformities of bees. It may make them fail to emerge from the hive, and that has a long-term life cycle effect within that colony. There's a few more details in a lot of detail actually on this section of the guide, so I'll just skip to the next one. So safety on farm, there's a little bit of detail on that, and I think if you're already a berry grower and you engage beekeepers on site, you'll have some farm safety procedures in place. And if you haven't already, I suggest that you put some real quality professional advice together for people visiting your farm, whether it's your own self or if it's your workers. Farm managers are responsible for complying with relevant health and safety legislation, like inductions for staff, training for staff, first aid, emergency procedures, etc. And it's really important that the requirements of those farm managers are met for the safety of beekeepers, for the health of hives, providing staff and contractors with appropriate workplace safety at all times. And another thing I wanted to mention that does come up in conversation ad hoc from the industry is to ensure that only beekeepers, auditors, or other agreed persons in your appropriate pollination contract have access to open or move the hives that are on site, so that there can be a better opportunity to manage the issues around that happening on your farm. Next slide. The other thing we mentioned in the guide is a little bit about native bees and pollinator-friendly farms. And I know Richard touched on this, and it's really important that, yes, native bees have the potential to complement honeybee pollination. And I've got some queries that I wanted to put through in the Q&A to Ben around feral bee densities. And we chatted just recently then about avocado farm hive density. So there's a great document that was produced by the CEO of AHBIC and some other fabulous people around hive densities in Australia, and it's published in 2022. And it actually refers to our industry and the stocking rates before Varroa. It was at a time when we were very hopeful that we would eradicate. And so even the figures in that document would detail stocking rates based on having some feral densities around the place. So the numbers of hives that will need to be available for pollination are definitely going to increase. But I think if it's anything to add, that yes, native bees can definitely complement honeybee pollination. Having a range of different opportunities for bees to forage on farm that are a range of sort of native species or supplemented crops that you might put in depending on the size and operation that you've got. This will actually help with managing honeybee health on farm. So one monocrop is definitely not what honeybees want or need, but it certainly goes a long way to ensuring the future of a healthy Australian honeybee industry by supporting some of these bee-friendly farming practices. So take a look at the bee-friendly farming practice guide that's provided by Ween Bee Foundation. So there's a link to that in the pollination guide itself. And that brings me to the end of my little snippet. I hope that was quick enough so that we can move on to...
Yes, we have actually run out of time, and I don't know if there's any questions from any of our participants. So hopefully that means they're just so overwhelmed with their new knowledge about bee bonking that their brains are full. But obviously there was a lot that we covered today, but I think both presentations and obviously your presentation, Bianca, were very comprehensive. And just the idea of this was to give everybody something to think about what the post-Varroa future might look like in Australia. And I think it's really important for those of you who are interested in that pollination guide. I have popped the link in the chat, but you can find that on our website. Similarly, we will have this webinar will be recorded, and considering the amount of focus on Varroa around Australia and the Varroa development offices, and we've just got a project in Queensland, there will be more and more resources coming out. But I think for me, the key message is things will be different, and we cannot expect things to go on as they have gone on. Obviously, Ben, I don't know. I know that you're Emeritus Professor now, so I don't know if you're still doing your amazing feral bee sampling or if you've handed that on to some Swedish acolytes. But yes, I do hope, obviously that was incredibly clever science, and how you did that was really amazing, particularly with the cigarette butts. But yes, and thank you, Richard. Obviously, getting an insight from someone who's actually been there, done that. I know for me, when we were doing the Varroa stuff, we heard from some New Zealand beekeepers, and that was when it finally clicked for me what this meant. So I really, really appreciate you doing your first ever Zoom call, I believe. So that's impressive.
It is.
Yeah.
It
And
is.
you've
And
done,
if you ever got another incursion like that, the best thing you can do is go out 100 kilometres and bait it with poison and work your way in. Because unfortunately, that's what we're having with the hornets at the present time.
yeah. Yeah.
Mm.
And look, we really, really thank you for your insights, Richard, and you get an A+ for your first Zoom presentation. Well done. And thank you, Bianca, for pulling everyone together and engaging with our speakers and bringing this together for the berry industry. We're really appreciative. And I hope that those of you who joined us from other industries found it useful. And I wish you all a very fantastic festive season, and enjoy lots of honey and fruit and avocados and all of those good things. All right. Thanks very much, everybody. Have a great day.
Bye.